Hello everybody. We are gonna give 1 more minute to wait some more assistance. Morning everybody. I am Jose Miguel, Bioprocess Product Manager in Condalab, and today we are going to talk about the application of microorganisms for the fermentation of active pharmaceutical ingredients in the pharma industry. First of all, I want to tell you that if you have any question, you can write them in the tool. So at the end of the meeting, if we have time, we will answer all the questions that I can. So with no more delay, I'm going to switch off my camera and then I will share my screen with all of you. So for this meeting, I will fragment this presentation in 4 parts. First, I will talk briefly about the origin of the fermentation process and some application. Then the workflow and impact over the pharma industry of the fermentation. We will follow with the pros and the cons using this method of synthesize. And finally, I will talk about some essential components to optimize this fermentation. From a biochemical point of view, the fermentation is a catabolic process that generates electron acceptors under anaerobic conditions. There is no oxygen, so the reducing power created during the glycolysis must be consumed to regenerate the coenzymes and maintain this metabolic pathway. However, if we speak in an industrial setting, we call fermentation to the synthesis of a metabolite using microorganisms. The production, we could also say the production of an organic molecule through chemical reactions of bacteria or yeast. Originally, the products obtained from the fermentation were foods such as bread or cheese, or alcoholic beverages such as beer or wine. But during the 20th century, the power of microorganisms began to be used to synthesize specific compounds. Here I have some examples. For the first one, at the beginning of the First World War, the British obtained acetone through fermentation using a strain of Clostridium. They used this solvent to produce explosives. Then about food or additives, there is the citric acid. it began to be marketed in the 19th century, and it was obtained from lemons. But since 1919, it has been obtained with fermentation through Aspergillus niger. And the last and best example for this presentation would be the synthesis of antibiotics. The most known is the penicillin, whose golden age started at 1945. According to the World Health Organization, an API, or active pharmaceutical ingredient, are the substance used in a finished pharmaceutical product to develop a pharmacological, diagnostic, treatment, or preventive effect over a disease. The API are the active compound of a medication, those with the therapeutic capacity offered by the drug. A pharmaceutical drug, a medicine, is composed by the API and the excipient, mistakenly called an inactive substance. But that's not true, because the excipient can fulfill specific functions to facilitate the action of the API. For example, it can regulate or modulate the solubility and the viability of the API, making it easier to be absorbed for the patient. Also, it can regulate the stability, the flavoring, so it will be easier to be taken, the coloring, and the emulsifying. Most companies in the pharmaceutical industry still relies on the production of small molecule APIs. There are other methods besides the fermentation. This could be through chemical synthesis, or the purification from raw material, commonly plants, vegetables. The chemical synthesis has been the common method. It has a great robustness and stability, but it is only applicable for the simplest molecules. Meanwhile, the purification method consists of extracting the API from the raw material that synthesize this molecule naturally. This method requires large plantations to maintain the production, since the yields are very low. Companies use this last method for molecules as last resource because there's no as viable as the chemical synthesis. But during the years, more and more microorganisms are being used for the synthesis of these APIs through fermentation. The first example was, as I said before, the antibiotics, the penicillin in 1945, but since then other products came. The options in the industry only grow. There is a tendency to use media and ingredients that generate no toxic waste, a difference of the chemical industry. So there is a less risk to the operator and the environment. The options of synthesize APIs through fermentation has been increasing thanks to the advancement of the molecular biology, which allow to understand different metabolic pathways, and the evolution of genetics, giving us the possibility of creating a genetically modified microorganism, a GMO, and then you can manufacture a specific molecule, the API you are looking for. Separately, There is a fourth way to produce medicinal drugs, that is through cell cultures or mammal cells, which are the main tool to synthesize complex drugs. However, working with microorganisms is much easier and more scalable than cell cultures, in part because the media also tend to be more affordable and microorganisms has a greater flexibility and a reasonable price. In order to produce an API through fermentation, a development phase is required in which the microorganisms is crucial. You have to select the specific bacteria or the specific yeast for this job. Then the research team have to formulate a media that enhances the growth of the microorganism and the synthesis of the desired product. The bioreactor procedure must be checked. You can use an stainless bioreactor or a single use. Also, you can schedule the manufacture times using a batch or a fed batch procedure. Then other necessary equipment has to be selected, like the supplements or buffer regulators of the culture. And finally, the process has to be optimized by calibrating each variable. Will be the exact moment of inducing the culture, the pH, the temperature. Once complete, it will be moved onto the manufacturing of the product, dividing into the upstream and the downstream. Here we have a resume of the upstream, the fermentation of the API, and the downstream, the purification of it. In the upstream, we have the sterilization and the strain preparation. Then we have a inoculum phase, where it is necessary to reach a specific concentration of viable cells to be able to take them to the fermenter. And the last stage of the upstream will be the fermentation stage, where the API is produced. Usually, it can be in batch or fed batch schedule, and the batch is a process where the necessary compounds are added at the beginning of the fermentation, and the fed batch is when the nutrients in deserts and others are inoculated through the fermentation process. The second phase is the downstream, where first you have to eliminate the microorganisms, usually by filtration. Then there is an extraction and purification of the API, which will be taken as an ingredient for the manufacture of the medicine with the excipient. But also, you have to eliminate the culture media, the waste, and other products from the bioreactor, and you have to clean it. So you can use in the next batch if it is a stainless bioreactor. If it is a single use, you use them and then you throw it to the rubbish. The fermentation are processes that last hours or days, but the purification usually takes more time. However, it is the development stage where the companies takes more time. It can take even years, and in several cases, the pharma industry still study how to optimize their processes while it is in their production phases. Here we see the main types of medicines that are produced, or at least their active ingredients, through fermentation. On the left, we have first the antibiotics, for example, the penicillin, the cephalosporin, macrolides, then different products that can regulate the immunitary system, for example, immunoregulators, usually with a polysaccharide structure. Then there will be the immunosuppressants. For example, we have here the structure of the tacrolimus. Sorry. We were talking about the immunosuppressants. For example, the tacrolimus. Here we have the structure of the molecule. And this product in specific is used to avoid the rejection in kidney and liver transplants. And on the left, we can see other therapies, for example, the RNA interference, usually for diseases like amyotrophic lateral sclerosis or spinal muscular atrophy. And also, you can produce oligonucleotides used as antiviral therapy through fermentation. Now we are going to see the advantages that is produced in this system, with that is system produced, and some drawbacks or challenge that must be solved. Comparing the characteristics of each production system, we can highlight that the following characteristics. Thanks to fermentation, we can obtain molecules that will be too difficult to produce with the chemical synthesis. And it can select, and we can use the fermentation to produce APIs that will be completely inaccessible or unprofitable with the chemical pathway. And regarding to the case of the purification, we avoid the requirement of fermentation. So we can reduce the process from years to weeks, avoiding the environmental impact of large crops. We obtain a higher yield, and we don't depend on the weather, and allow a greater flexibility in choosing the location of the industry. Also, we gain the possibility to produce different ingredients, because with the fermentation method, we only can produce the APIs that these plants produce naturally. But with GMO organisms, with genetically modified organisms, we can use the bacteria to produce even more APIs. That doesn't mean that the fermentation is a hassle-free process. The main problem is that you are using living beings and raw materials, raw ingredients. So you are using roast and generating viability. It should also be noted that it is a laborious process, both in the initial calibration, in the development, and in the manufacturing process. The material required are specific, and the personnel must be qualified. Also, you have to guarantee an sterility to avoid the competition between microorganisms, because the presence of a non-invited microorganisms reduce the amount of available nutrients and reduce or even inhibits the growth of the desired bacteria. In these croquis in this table, we can see a summary comparing the three exposed methods. The chemical synthesis is the most stable and robust method, and is also show a high yield, but it only gives the simplest compounds, and it's limited, and it is a limited process and difficult to find new therapeutic molecules. So industry use massive screening looking for new candidates, but this screening has a low possibility of finding new available APIs. In purification, you can produce complex ingredients, but the production, the yield of production is very low, and you have the problems associated with the crops with a big plantation. Finally, the fermentation method is the most flexible because it allows the production of a variety of products beyond what chemical and purification allows. However, it has a greater variability and requires a high specialization. In our last part of the meeting, I want to show you about the different ingredients and culture media used for the synthesis of active molecules. The selection of a culture media depend on the chosen microorganisms. Here we have some culture media used in the synthesis of active molecules. For example, we have the IPD broth used for the expression of proteins in yeast, the PPLO broth base without strain, without crystal violet. It is used for mycoplasma. Then the terrific broth and the LB broth, in specific the Lennox broth. They are used for E. coli to synthesize proteins or enhance the growth. Then the reinforced Clostridium broth for the Clostridium growth. And finally, the MRS broth for Lactobacillus. But beside of the culture media, there are what is called bioingredients, components that can be used for the formulation of the culture media or as a supplement to improve the growth and the synthesis. We have to differentiate these bioingredients from other supplements that help us to avoid contamination, for example, antibiotics, or others that select the specific microorganisms. These methods can affect the yield of the production, so it is preferred to avoid them and work in sterile environments since the preparation of the culture media and the inoculum since the beginning. Speaking again about the bioingredients, they act as a nutritional contribution for the culture media, being a great source of carbohydrates, nitrogens in form of amino acids and small peptides, essential vitamins, and others. Some examples are the dextrose, the saccharose, hydrolyzed or extract products. One of the most popular bioingredients for fermentation are the peptones. These are protein hydrolysates that promote the growth and specifically the synthesis of metabolites. In the case that you want to stimulate the growth of the bacteria, you can use extract like the yeast or malt extract. And just as a curious note, previously we mentioned about the synthesis of complex drugs, the use of culture cells of the cell cultures. It is an alternative method to the fermentation using mammalian cells instead of bacteria or yeast. And this method gives the ability to produce even more complex therapeutic molecules. For this culture, there are bioingredients, for example, peptones with a low level of endotoxin. And here in Condalab, we have a line called Condalo designed for the synthesis of biological drugs with mammal cells. Finally, I would like to summarize and draw conclusions from this talk. First, I will emphasize the ability of microorganisms to produce active molecules with therapeutic power through fermentation. Secondly, I will indicate that the fermentation allows us to find medicines that will otherwise be inaccessible, either due to the inability or being too high to produce with a high cost. And finally, the importance of the ingredients and culture media to obtain the desired amount of metabolisms, the desired yield of API. Besides, if you want to know more about the pharmaceutical industry or the synthesis of products through cell cultures, I recommend you to visit our Condalove Talk section in our website, where you can see other webinars. And an other option I recommend you is to download our bioprocess brochure, where you will see more culture media and some bioingredients for the manufacturing part, but also this brochure includes the methods for microbiological quality control. Thank you for your attention, and I hope you find it interesting. At the last, I will answer your question. So I will open the panel, and first question I have is, what other contribution can bioingredients provide? For example, the peptones, besides being a source of nutrients, have a buffer effect. They generate an environment where the APIs produce and secrete to the culture media do not suffer any alterations, specifically if they are protein-based, if they are structure based in a protein. Other question is, does API apply to biological drugs? I will say that technically yes. For example, a biological drug is a monoclonal antibody, and this molecule is an active ingredient with a therapeutic power. So it can be considered an API. However, biological drugs are obtained through their cultures, not through microbial fermentation. So I would say it is an API, but synthesized in a mammal cell like CHO or HEC instead of a bacteria. I have another question here. It may require it may entire microorganisms be it may entire microorganisms be the API. If the whole microorganisms can be the API. As far as I know, no, because as I say, API active pharmaceutical ingredient, the whole microorganism is not an ingredient. But I know that they can be used in specific situations, such as probiotics or in microbiota transplant. So in this both case, help with intestinal diseases. Other case different will be the use of microorganisms to produce vaccines. But as far as I know, I will not consider the whole microorganisms as a API. And I see we have more questions, but we are running out of time. So we will send you, all of you, the record of this presentation, and also we will answer the remaining questions, so none of them will be abandoned. I hope you find this talk interesting, and thank you for coming. Have a nice day.